Hello everyone, welcome to the third objective and the final objective for chapter 16. In the previous two objectives we talked about the structure of DNA, then we talked about how it was replicated, and now we're going to talk about the genome organization, how it's packaged together. Okay, so as we begin to talk about organization of our genome, we're going to go from smallest to largest. And we've already talked about this, quite a bit in fact, is how the nucleotides will bind to each other by phosphodiester bonds, and then how they will form hydrogen bonds with an adjacent chain of nucleotides. And if I were to draw this in the form of a double helix, it would look something like this. I'm limited on how large I can draw this simply because the slide page is not that large here. But this is our DNA then, that we're used to thinking about. So the smallest form is going to be the nucleotide. And then as those nucleotides are put together, they form DNA. And this DNA then is now organized into genes. And so let's block off a segment of this DNA. And let's call this the gene. So what is a gene? Well, we're going to come back to this term throughout the semester. But for now, what you should know about a gene is that it is a sequence of DNA that usually codes for a protein. This protein coded for by that gene is what gives that cell or that organism its function. And as I said, this is of course just a brief schematic, but recognize that we're talking about millions of nucleotides here in a row, all this DNA just stretched out. In fact, if you were to take a single cell from your body and crack it open and take all the DNA out of that cell and stretch it end to end to end with all the DNA in your cell, you would have roughly 2 yards. Or you could say 2 meters or 6 feet. If you have 2 yardsticks at home or 2 meter sticks, take them and put them together and look at it for a good visual. That's how much DNA we have in each of our cells. To illustrate this point, let's ask the question, if we took every single cell in our body, all trillions of these cells, and cracked them all open and took all the DNA out and stretched it end to end to end, how much DNA would that be? How far could we stretch it? Well, I'll just tell you. If we draw our good friend planet Earth here, and we put all of our DNA together from a single person, all the trillions of cells they have, it would stretch all the way from Earth to the planet formerly known as Pluto, and wrap around Pluto and come all the way back. And when it got back to Earth, there'd be enough DNA to go probably back to Saturn. That's how much DNA you have in a single cell. That number always impresses me. The question now is, how do we package all that DNA into a single nucleus? Seems like quite a feat to take 2 yards of DNA and put it inside a tiny nucleus. I often make the analogy to if you're packing to go on a vacation and you have more clothes than you could think of that would actually fit into your suitcase. Instead of taking those clothes and cramming it in there and barely being able to shut it and probably wrinkling all your clothes and it not being very usable, what you've probably learned over the years is that if you take your clothes and you roll it up nice and tight, you're able to get a lot more clothes into a single suitcase. Well, interestingly, that is exactly what your DNA does when it packages your DNA into the nucleus. Let's see, we forgot to put the number 2 here, so let's do that real quick. And then let's move on to how we package this DNA. And this next level, we're going to call chromatin, the level of the chromatin. And I think the best way to illustrate this is let's draw a straight line here. And this straight line is our DNA molecule. It's this double helix, but it's just to illustrate how large it is. Now, what our cells will do is they will use these proteins called histones. And so let's draw some histones. They usually exist in a structure of 8 histone proteins. But for simplicity in drawing, I'm just drawing 4 of them here. And we called these histones. So let's go and write the word histone here. In fact, let's go ahead and draw a couple of these structures. And remember, these histones that I'm drawing here, they're proteins. And what will happen now is this large stretch of DNA will wrap around these histones. So let's go ahead and just show this like so. Something like that. And in packing it this way, you're able to get a lot more DNA pushed together. So chromatin, a good way to define chromatin is to say it's a combination of DNA plus protein. And that's what your chromatin is. It's often difficult as you're learning this to distinguish the difference between chromatin and chromosomes. And we'll try to do that a little bit in this chapter as well as the material on Friday. So at this stage of chromatin, we're still able to package a lot of DNA together and get it in that nucleus. But we still have level of DNA folding. And that's the level of the chromosome. And that would be taking these structures here, and I'll do my best to draw this here, at the level of the chromosome. And all the chromosome is the exact same thing as the chromatin, except it's further condensed. That means further pushed together, further packaged. So if we take the structure of the chromatin with the histones, and what happens is it gets pushed together. This is the best way I have to draw it. It's pushing these all together, and I guess we can draw some blue dots for the histones, just to let you know that they're still here. It's just folded upon each other, like so. So again, chromatin is the DNA plus the protein, and chromosomes is when you take the chromatin and you fold it upon each other until you get a really constricted structure. Condensed structure would be a better term for that. There is one term I forgot to define, and that is these histone proteins, when they are packaged up with DNA together. So the histone proteins and the DNA are called the nucleosome. Alright, now we have one more level of our genome organization to think about, and that is the genome itself. And the genome itself, we can think of it in many ways, but I think it's going to be better to show a picture here. Sometimes when you hear the word genome, somebody might think of all the single letters of our entire DNA makeup. And that's not an inaccurate way to say, because that's what it really is. You know, the genome is all of the genetic material of an organism. So you could think of it this way. But let's look at it in this sense here. These are our chromosomes. In fact, that little squiggly thing I typed back there, or drew right back on the previous slide, it was the chromosome. And this here is also a chromosome. As is this one. As is this one, and we can keep going down the line here. In fact, sometimes, let's just pick one here. Let's pick chromosome 5. It's a good one to look at. Remember that the chromosome is nothing more than a large piece of DNA. So if I were to draw down here, this is what your chromosomes are. It's just a large piece of double-stranded DNA that, through the packaging of the histone proteins, condensing it, and then condensing it even more to form this very constricted, condensed molecule called the chromosome. So the genome is the collection of all of our genetic material. That means all of these chromosomes here together make up our genome. So the genome is the largest part of the genetic packaging in our cells. We're going to come back to this picture, maybe not this exact picture, but we'll come back to this representation of our genome. In fact, what it's called is a karyotype. That's not a bad word to kind of put in your memory. This comes from a Down syndrome patient. That's not particularly important right now. We'll come back to that in a little bit. As you've probably discovered throughout this chapter, through these 3 podcasts, there's a lot of new terms for you, and it's super easy to get confused with them. 2 hints I can give you is to write these structures out. Write them out several times. If you have a whiteboard, in your home, in your dorm, in your apartment, wherever you study, in the library, just write these things out over and over again. Another good strategy is to build some note cards with these different structures on a nucleosome. On the other side, define it. Histone, on the other side, define it. That's probably the best way to learn these terms, because there's a lot of new terms here, and it is quite difficult to not get them confused. If you have any questions as you're going over this material, please let one of us know, and we'll be happy to answer any questions you might have. But we'll see you in class.